tell you a little bit about sort of molecular biology that we've been doing down the road from here at the MRC Laboratory of Molecular Biology. So one of the sort of homes of molecular biology. But yes, molecular biology sort of just jump straight in is a bit heavy, so I'll try and tone it down a bit. And please, please put your hand up or just yell out if there's something that's a little bit, bit sort of past over you. And maybe it'd be sort of interesting to talk about whatever you find, a bit baffling. So please speak up if it's a bit much. So in previous years, we've sort of we sort of told you a little bit about who each of the speakers are. So we've sort of got people at various sort of career stages who you can hear from. So my stage is I'm a postdoctoral fellow. So I've been through undergraduate. I was an undergraduate down at Imperial. Did a PhD down in King's College London, and then I came to Cambridge 5 years ago as a postdoc. So I'm at the point where I've sort of done sort of nearly 2 postdocs now, and I'm at the point where I'm sort of starting to look for maybe getting my own sort of research group. So this is the sort of kind of career progression you're looking at if you think about doing science. So I'm going to tell you sort of call by talk, rewriting the language of life. I'll tell you what that's all about in a moment. But I just wondered has anybody ever heard of this area of biology called synthetic biology? Maybe show of hands. Have you heard of it? Put your hand up. Yeah, just a few. That's good. Okay, so this is kind of what it is. So there was a recent paper looking at sort of exactly this, sort of how many people have really even heard of this, and those numbers are about right. So it's only just sort of getting a bit of interest. But hands up if you've heard of genetically modified organisms. Everybody. Okay, so synthetic biology includes all of that stuff. So it's really, the reason why I put the sort of scare quotes on new is because, in a way, the idea of synthetic biology is quite old. It's really born out of sort of synthetic chemistry. So in synthetic chemistry, this is where chemists try to understand precisely the sort of nature of a molecule. And if you think you know what a molecule looks like, draw the structure on paper or whatever, then you ought to be able to synthesize that in the chemistry lab. And then if you find that when you've made what you think is sort of a synthetic version of this chemical, you can test it out. And if it turns out to have exactly the same properties in every possible assay you can think of, then you've pretty well understood, and you're very likely that you've got the structure right. Okay. So in the 1920s, a chemist suggested, can we potentially do this with biology? Biology really is just sort of chemistry showing off in a way. So can we start to think about, making pieces of chemical systems that approach life? Maybe don't go straight towards, exactly, recreating life. But can we sort of look at aspects of life and recreate some of that, or recapitulate that in the test tube? Okay. So some of that includes looking at existing biology, like crops or bacteria, and tinkering with parts of it, sort of putting in sort of new sort of circuits and so on. And that includes, so that's one of the sort of branches of synthetic biology. But one of the others is what I sort of call myself, which is sort of a bit more sort of towards the fundamental side of synthetic biology. So do we sort of understand some of these aspects of life well enough that we can build them? So we've really been driven by looking to the kind of what are the kind of really key hallmarks of life, and the sort of the best definition we have of life is that it's a system for performing heredity. Okay, so the transfer of information. So really, why you have aspects of your sort of traits from your parents, and your offspring will have some of the traits from you. So there's a flow of information. And also Darwinian evolution. So some of that information is slightly changed during each sort of step in each generation. So these two kind of hallmarks really define life. So can we, as synthetic biologists, come in and make systems that are capable of this synthetically in the laboratory? Okay, so can we think of sort of alternative languages of life? Can we have something that's sort of a little bit like a sort of Rosetta Stone? This is a sort of an Egyptian relic that has sort of 3 different scripts on it, and sort of allowed scholars to understand the language hieroglyphics at a time when They didn't know how to interpret them, but they understood Greek and Demotic to a certain extent. So the discovery of this relic was sort of really the key to opening up this kind of language. So can we do something similar? Can we sort of get some real insight into life and the kind of chemical boundary conditions? So sort of what kind of chemistries and properties are the necessary ingredients that are going to give you life? Can we do that by thinking about sort of alternative chemical languages? Okay, and really sort of more generally, this is of interest to sort of fields like astrobiology or fields like origin of life work. But beyond sort of the kind of knowledge economy, we think these sort of things really ought to lead towards the development of novel tools and technologies for research and for medicine. I'll show you why that's the case, hopefully at this time. So very central to biology, hopefully you've maybe seen this before somewhere, this is called the central dogma of molecular biology. And it's, this was largely dreamt up by researchers like Francis Crick, who used to be in the MRC labs. And it really describes the flow of information in biology. So of course, we all know information is stored in your genome in the form of DNA, and this can be read out as RNA and then translated into proteins, which are then sort of workers that perform the job of building cells and sort of setting up the whole system to allow the replication of DNA. Okay, so we've been really doing over the last few years a certain amount of work to try and build a sort of third nucleic acid system, or fourth, or fifth, or sixth. So really kind of separate but connectable to the central dogma. Okay, and these are informational polymers that are distinct chemically. So they're not DNA, they're not RNA, they're not protein, but we've been exploring these for the storage and propagation of genetic information, okay, or synthetic genetics. Okay, and these fall under this kind of umbrella term of XNA, or xenonucleic acids. Okay, so really looking to the future, we could imagine kind of genomes or simple kind of replicating circular entities within cells, which allow you to sort of have a kind of firewall that walls off genetic information from natural biology. And there's a sort of good argument that this is a sort of potential way to ensure that synthetic genes, if you make them sort of genetically modified organisms, can remain separate and distinct from natural biology and can't be inadvertently replicated or sort of escape. So there's a sort of there's that element to it, but also I was going to try and get somebody to come out and draw this, but if I say to you sort of DNA or RNA, sort of hopefully probably the first thing that jumps into your mind is a double helix. Okay, but really the double helix is one of the sort of in a way, this is a bit heretical, I probably shouldn't say it at Cambridge, but in a way it's one of the most boring things you can do with a nucleic acid. Okay? And if you take a double helix and you peel it apart into the two separate strands, each strand will fold upon itself into a well-defined structure. Okay? So you might have, you might be sort of aware of this if you've done things like translation in biology lessons, where you've studied, say, a machine called the ribosome. So this is the molecular machine in your cells. You've got about 100,000 of them in every cell, which will take an RNA message and read it out and make the protein that's encoded within it. Okay? Well, the ribosome is actually a machine constructed of some proteins, but mostly at its heart, it's RNA, so peeled duplex apart, and then one strand folded up. Okay? So although it sort of looks like nucleic acids sort of want to be a double helix, if you bring sort of circumstances together that will allow a single strand to be made, that single strand can fold up into really a kind of bewildering variety of different structures. And some of these can be really sophisticated machines or even sort of sensors. Okay? So can we do something similar to that? Can we start to think about not only replicating information, but doing things like making single-stranded structures and machines and so on out of XNAs, alternative polymers that nature doesn't use. So this is a sort of deliberately kind of horrific looking chemistry slide, just to show you that. So here at the centre, you may or may not recognise this, but this is the sort of building block of a piece of DNA or a piece of RNA. This one's DNA. So this is a DNA nucleotide, or rather nucleoside, whatever like that. So DNA building blocks are composed of a phosphodiester backbone, okay, linked to bases, nucleobases, A, T, C, G, and the backbone and the base are linked by a sugar, okay, a sugar called ribofuranose. Okay, now this slide just sort of shows you that there really is this kind of bewildering variety of modifications to any of these sort of three components of a nucleotide. So you can make nucleotides with different backbones, you can make nucleotides with different sugars, you can make nucleotides with no sugar, or with a different kind of chemical entity connecting the backbone and the base. Or you can even move to totally different bases. Okay, different molecules, functional groups that are capable of doing the same sort of job that a base does, but not a base. Okay. There was a recent sort of news report where a team in the US has sort of managed to get a single non-natural base pair growing in a bacterium. Okay, so there's really a huge variety of kind of changes that you can make. And each change that you would make, if you pick any one of them, so you sort of, let's say you keep natural bases, but you change the sugar, okay, that gives you a polymer which is an XNA, okay, it gives you a polymer which is not a natural one, okay. Now, why would you ever want to do that beyond sort of, say, just interest? It's hopefully going to be illustrated in the next couple of slides. So here's an XNA that we've done probably most work with so far, which is called HNA. Okay, it's called HNA because instead of the ribofuranose sugar connecting backbone and base, you have this sort of thing. This is a sort of instead of this sort of 5-membered ring, okay, you've got this 6-membered anhydrohexatol. So that's not actually a sugar, it's sort of a different chemical conjugate. Okay, now if I start to move these off, you might think, well, actually, doesn't it sort of vaguely still resemble DNA? And yes and no. But really, in the chem lab, this is one of the most drastic changes you can make. It affects the polymer, the duplex, in really quite a distinct way. You can sort of see it's got this kind of hole running down the centre, so it really doesn't look like DNA at all. It's got this kind of really extra twist to it. And the sort of the chemical nature of the backbone, the way the charges are displayed, the sort of how kind of oily it is, hydrophobicity, are really very different between these two things. So these are really very chemically distinct entities. Okay, and then here's the kind of here's one of the kind of things that gets us kind of excited about using these. Okay, because their structures are very different, their properties are very different. Okay. So with XNAs, you're potentially getting really very useful properties that you could maybe exploit in medicine. So in the case of HNA, if we just take a polymer made of HNA and we pop it into blood, okay, into 100% blood serum, okay, so your blood is full of protein enzymes that are designed to slice any foreign DNA or RNA to pieces, okay, called nuclease enzymes. Now your body does this because it's very keen to keep its DNA and RNA in the right places. Okay, and generally, if you've got DNA floating around in your blood, it's probably not come from you, or it might have come from, say, a cell that's died, or it might have come from a virus or something. And so your body wants to destroy that as quickly as possible. Okay, so if you pop RNA into blood, it's gone sort of within minutes. If you put DNA into blood, just kind of inject you. Within sort of half a day to a day, it's pretty much destroyed. Whereas HNA, you can go on for several days at least, and we've done some work that shows it goes even further than this. But it's essentially untouched by these nuclease enzymes, partly because it's so very different in structure. So if we were imagining making machines, nanostructures, sensors, this kind of thing, tools out of HNAs or out of XNAs, we could immediately make them, or the prospect of making them, so kind of robust that we could use them as tools and medicines, and they would survive within your blood long enough to have their sort of desired effect. Okay, so how long have I got? 10 minutes? Yeah. So one of the kind of really powerful phenomena that we want to seek to tap into is Darwinian evolution. Okay, so if we have systems that allow replication of polymers, so just as your body does with your DNA, and it's not entirely perfect, there's sort of replication errors, and then you select out the sort of the desired properties you want, this immediately sort of allows evolution to occur. Okay? So if you say you want to make a drug which will switch off a cancer cell, kill it, you could potentially do rounds and rounds of evolution towards that activity of killing. Okay? So to be able to do that, what we need is mechanisms for replicating XNAs, and that's really what our work's been focused on. So I'm going to skip over a lot of details, but we were able to take enzymes that are usually, whose usual job in nature is to copy DNA, okay, and it happens, so skip over, but using enzymes, polymerases that come from an organism growing close to one of these sort of deep sea volcanic vents, we isolated sort of a polymerase and then performed sort of test tube evolution experiments to change its specificity. So enzymes are, you'll have done this in your lessons, enzymes are like sort of locks, and their substrates are like keys in the sense that they're very specific, so only some substrates will be recognized by an enzyme. Okay, and polymerases are no different. They recognize DNA, they recognize RNA, but they don't recognize unnatural things like XNA. Okay? So if you want to copy a piece of XNA, a natural polymerase generally won't do it, or won't get you very far. Okay? So we were able to do rounds and rounds of evolution in the test tube to get our polymerase enzymes to accept XNA building blocks. And along the way, we discovered lots of interesting things about the way polymerases work. I won't go into that. Okay, so just to sort of almost repeat myself, doing these sort of directed evolution experiments on the polymerase enzymes then allowed us to get sort of the two properties that we would need to take a piece of DNA and then copy it into XNA. So use the DNA as a template. So we give it, so we have this first polymerase activity where we give it XNA building blocks and a DNA template, and it will make a copy of the DNA by reading along and putting an XNA building block where sort of exactly like regular DNA copies. Okay? That gets us one way where we can then we can sort of make XNAs with sort of sequences of our choice. We then came up with again polymerase mutants that we evolved in the lab to then take a DNA template, and then when we take an XNA template, and then when we give this one DNA building blocks, it will copy it backwards into a piece of DNA. Okay? So then that allows you to tap into lots of tools and technologies that are very standard in the lab for looking at DNA, like cloning and sequencing. Okay? So altogether, we've got about 8 or 9 different XNA chemistries which we can do this with. So we can take DNA, we can copy it into XNA, have it challenge it to evolve itself. Okay, so challenge the XNA to become like a ribosome, although not that complicated. Become a kind of artificial enzyme or an artificial sensor, and then when we want to know what sequence it's adopted, we can make that back into DNA and sequence it. So I'll largely skip over this, but just to sort of show you very, very, very quickly in the last closing minutes the sort of thing that we do. I mentioned sort of single-stranded nucleic acids give you interesting properties. So here is, just to kind of really underline that point, this blue thing down here is a short piece of RNA selected in the lab, and this brown thing is a protein, okay? So maybe if I just play that again. I just wanted to kind of underline for you that even though this thing is composed of merely 4 different chemicals, it's building blocks, okay? It's capable of making a really, really sophisticated kind of interaction interface with the target molecule that it was evolved to bind to in the laboratory, okay? So, in other words, nucleic acids can actually be capable of as sophisticated kind of molecular functions as proteins. So I will more or less skip over this, but essentially we came up with mechanisms using our mutant polymerases that I told you about to copy DNA templates, challenge XNAs to fold up and bind to targets of interest, okay, recover them back to DNA and sequence them. Okay, and then doing this over and over and over, we proved that the XNA is capable of supporting evolution. Okay, so we can see the sequences of the XNAs indirectly by bringing them back to DNA, and we proved that these things over time will evolve to get better and better at hitting targets of our choice. We've got lots of fancy techniques for measuring how well one molecule interacts with another molecule, and when we look at the XNAs that we've evolved in the lab, we find that they get better and better at hitting their targets. So it's a bit like having a key that gets better and better at fitting into a lock. Okay? So XNAs can not only perform evolution, heredity, but they can also perform evolution. And most recently, what we've done is, I'll skip this, I guess. Most recently, what we've done is, a bit like a ribosome, we've managed to select out XNAs which can recognize an RNA target and will cut it. Okay, so it's a bit like a sort of molecular scissors. So, and it's a bit like a little molecular machine, but it's composed, and we've done this in sort of 4 different chemistries, but it's like a machine but composed of an entirely artificial backbone. And then maybe the very last thing before I just sort of summarize everything. Once we sort of found we could do one activity, cutting, so making effectively a kind of artificial enzyme out of XNA, we wanted to find out whether we could do something sort of even more sophisticated. And that's where we selected, again, in sort of directed evolution approach, for an XNA enzyme, which, when we give it two different XNA substrates, so this is one XNA with 2 XNA pieces, okay, it will stick these together, it will assemble them, and when we cut that piece of XNA out and get rid of that enzyme, that piece of XNA will fold up and fold itself up into a second enzyme, okay? Which, when we give that second enzyme its RNA substrate, it will cut it, okay? So this is one XNazyme assembling a second XNazyme which chops RNA, okay? So this is really kind of a sort of super simple compared to what nature's capable of, but this is something approaching a sort of metabolic pathway. It's in metabolism, which you'll learn about in painful detail, life is really all about catalysts assembling other catalysts to then perform jobs which allow the whole cell to survive. So here we have a really simple version just from doing test tube evolution experiments where we've proven that the sorts of properties that life can have can be performed with fully unnatural polymers. So just to kind of repeat some of that back again, XNAs are a wide range of artificial polymers, okay? They've got lots and lots of different properties, so you make lots of different modifications, you get different properties. Some of these are very useful properties if we're going to seek to use these for research and medicine and so on. Now, we've made systems out of evolved polymerases that then prove that these XNAs are capable of forming genetic systems in themselves, okay? So they can support heredity and evolution. And that really opens up what's called chemical structure spaces for evolutionary exploration, and potential sources of tools and technologies and therapeutics. So I've shown you very quickly just sort of some of the proof of principle work we've done to prove that these XNAs are capable of sort of what's called ligand recognition. Okay, so they're able to kind of recognize other molecules if you evolve them to do so on a really kind of atomic precision level. And most recently, we've proven that you can even get them to evolve to form enzymes. So really kind of some of these kind of really key features of life seem to be an emergent property. They're not simply, it's not sort of something special about DNA or RNA or proteins. It looks very much like a range of other chemistries are capable of performing the functions necessary to give you life. So that really kind of offers a kind of exciting prospect for the use of XNAs for maybe building kind of alternative biology. And can we come up with systems, can we take this further to the point where we can make simple, maybe artificial cells, maybe systems within living cells capable of walling off genetic information, maybe leading to sort of safe GMOs, this kind of thing. Or even just kind of looking simply, can we build life from the ground up? Thank you.